All right, it is 2. So let's get started. Reminder that test 2 scheduled for Friday. This not on test 2, but it will be on test 3. As always, make sure to check the test folders on Blackboard to see what goes to what test. And it's going to be like the same as test 1 was for after you. Same amount of questions, probably same type of questions, short answer, multiple choice, all that good stuff. But that's not until Friday. So let's actually talk about what we're going to cover today. And we're going to continue talking about hemoglobin. Hemoglobin is very well studied, and it serves as a model protein for us to understand ligand binding and cooperative binding. So just a little bit of a refresher about hemoglobin. It is the molecule that carries oxygen from the lungs through your blood and delivers it to your cells. And hemoglobin is found in your red blood cells. Red blood cells are mostly just packed full of hemoglobin. And hemoglobin has 4 different subunits. Alpha 1, beta 1, beta 2, alpha 2. So that's one protein made out of 4 different subunits. And each subunit can bind oxygen. And they have something called cooperative binding, positive cooperative binding, where if one subunit binds oxygen, that makes the other subunits want to bind oxygen even more. And so we're going to look at how that cooperative binding actually occurs in hemoglobin today. So the first thing that we need to understand when it comes to hemoglobin is that it has 2 different conformations that it's in. The tense state and the relaxed state. So tense, or the T state, is when you don't have oxygen bound, so without oxygen, while the relaxed, or R state, does have oxygen bound. And this nomenclature of T and R, that's found in a lot of proteins. It's usually just tense, you don't have the molecule bound. Relaxed, you do have the molecule bound. So let's look at how binding oxygen can cause, or rather binding oxygen in one subunit can cause the other subunits to want to bind oxygen. So let's look at this picture. Here, blue is tense, red is relaxed. So just like in your blood, if your blood has oxygen, it's red. If it doesn't, it has blue. So we're going to be focusing on the blue first. And here in this little pocket, that's our heme. If you remember, the heme is the porphyrin ring, and it has an iron in the middle of that ring with a histidine, the distal histidine, and there's another histidine below it called the proximal. So when oxygen is not there, so we're going to, in the tense state, we're going to just say oxygen's not there yet. So that's gone. The iron, the blue is iron, that's out of the ring, and the ring is kind of buckling a little bit. And like I said, this iron is interacting with a histidine F8, and it's called F8 because it's on the F helix and it's amino acid number 8. So his F8 is interacting with this iron. And this histidine is connected to the whole other helix in our protein. So this blue cylinder is just a bunch of amino acids that's very simplified. Now, when oxygen binds, so oxygen is now there. Remember, oxygen has a lot of electrons. So what happens is that the iron comes closer to oxygen. So the blue, right? If you're following the blue to red, that's what I'm talking about. We're now in the red state. Iron comes closer to our oxygen. The porphyrin ring changes its conformation. So now it's in a plane instead of being bent like that. But most importantly, when that iron moves, the histidine that is interacting with the iron, histidine F8, also moves by 0.6 of an angstrom, or 0.06 nanometers. So it doesn't move that much, just 0.6 of an angstrom. But remember, it's connected to all of the other amino acids in the F helix because of the peptide bond. So all of the other amino acids get dragged as well. They move as well. They all shift or go through a conformational change. And this kind of sets off a chain reaction. So remember I said that it's made out of 4 subunits. So let's say one oxygen binds an alpha 1. How do the other 3 subunits know to bind oxygen now? Or why do they want to bind oxygen even more? Well, this shift happens in the F helix, and this shift causes some changes where the alpha and beta subunits meet. So in the T state, here we have an aspartic acid on the beta subunit interacting with a threonine, or sorry, a tyrosine on the alpha subunit. Oxygen binds, the helix shifts, and so you have a big shift in the conformations. Now, what happens between the T state and the R state, the whole alpha subunit shifts down. So now you have a aspartic acid interacting with an asparagine. So it's kind of like a molecular switch that is happening to convey to the other subunits, oxygen is around, adopt the R state. This image above, it's the same cartoon as I'm showing down here. The only difference is we're showing where the electrons are now. That's the only difference between these 2 pictures. You also get a change in the termini. So here we're looking at where the alpha 2, alpha 1, and beta 2 subunits are all coming together. You can see they have a very complex interaction. And that can change with the binding of oxygen as well. So when oxygen binds, the key takeaway is when oxygen binds, the iron moves closer to the oxygen. This causes the distal histidine to move. This causes the helix F to move, all the amino acids in there. That changes the interaction between the subunits, which will cause the subunits to now want to bind oxygen more. So in general, that is what's going on. And that is how we can explain molecular cooperativity, or sorry, positive cooperativity on a molecular level. By looking at the actual interactions of the amino acids, we can explain this phenomenon that we see in hemoglobin. When one oxygen binds, it wants to bind other oxygens, the other subunits do. So is there any questions about that explanation? Place where you got stuck at, a little confused, or anything you would want me to go over again with that explanation? All right, if not, we can move on. And so here's just basically the summary of what I just said in nice little words. So hemoglobin, tightly coupled, you have large tertiary changes in one subunit leads to a quaternary change in the structure. Remember, tertiary structure is the 3D conformation of one subunit, while quaternary structure is the change of the whole complex. So if you change the 3D structure of one subunit, you change all of the subunits. And hemoglobin only has two forms, T, which is without oxygen, R with oxygen. And you switch between these two. When a switch happens, the other subunits can feel that between the interactions of the alpha and beta subunits. T state does not want to bind oxygen because iron is further away, while R state does want to bind because the iron is closer to the binding site. When you bind oxygen, iron is pulled towards that oxygen, which causes the F helix to move, alpha and beta interactions change, C terminus change. All subunits can now go to the R. You have higher affinity for oxygen, positive cooperativity. So there it is again, nice laid out in words in case couldn't quite follow what I was saying with the pictures. So let's begin our first question here. So myoglobin looks a lot like the beta subunit of hemoglobin, which is what we're showing right here. So here's the beta subunit of hemoglobin, HB. And that's what MB, HBA, HBB stands for. Myoglobin, hemoglobin subunit A, hemoglobin subunit B. So let's imagine that we took the beta subunit of hemoglobin, removed it, and instead replaced it with myoglobin. Do you think if I did that, the molecule would still have the levels of cooperativity that we see if the beta subunit of hemoglobin was still there? Well, keeping in mind, if you look at the two, they look very similar. And here are the sequences and the important interactions. A lot of them are highlighted here, but that's basically what's going on. So do you think we would still have the level of cooperativity? Anyone have any thoughts about this? Because you still have a distal histidine right here. So any thoughts, or if you don't have any thoughts, any questions? You can always send me a private message in case you don't like being known that you speak up. Well, if no one wants to speak up, I guess I'll answer it. A lot less fun that way, but that's okay. So no, it would not, because you need all those amino acids there to have the same type of interaction, right? And even though myoglobin looks very similar to hemoglobin, because they basically have the same-ish function, it's very, very unlikely that all of the amino acids, all the critical amino acids that would need to line up to have that cooperativity would line up in the exact positions that they need to. So no, you could not just put a myoglobin in there, because it does not have the correct amino acids in the correct positions to have cooperativity. So that's the answer to question 1 there. So this one, actually I have a poll for. All right. So here's to test our understanding of the R and T states. So there are a ton of known mutations for hemoglobin. So here's to Yakima in Kansas. So in Yakima, C99 is mutated to a D. This stabilizes the R state. So is P50 higher or lower than normal hemoglobin? And in Kansas, you stabilize the T state because you mutate an N to a T. So is P50 higher or lower than normal hemoglobin? So this tests a couple of things. 1, do we know what the R and T state is? 2, do we remember what a P50 is? So I'm going to put that poll up, take a stab at it, and I'll be back in a minute once we have some people answer to go over the answer. We'll do about 50 more seconds. All right, let's take a look at this. So here's the logic, right? I stabilize the R state. If I'm in the R state, my affinity to bind oxygen goes up. So in the R state, you want to bind oxygen more. P50 is the concentration of oxygen needed to be 50% bound. So if I had 100 hemoglobins, P50 is the pressure of oxygen for 50 of those hemoglobins to be bound by oxygen. That's P50. Therefore, if my oxygen affinity increases, P50 goes down. Because if I want to bind oxygen more, it takes less oxygen to be 50% bound, because it's easier to do. while in Kansas, I'm in the T state. So my oxygen affinity is decreased. Therefore, my P50 is increased. If I don't want to bind oxygen, you need to put more oxygen into the system for it to be 50% bound. Right? So that's how R and T states, if you're forced to be in one like you are in these mutations, that's how they can affect the P50. So questions about that idea or anything we've talked about up to this point? All right, then we can move on. Let's talk about ways that oxygen binding of hemoglobin can be changed. First, we're going to talk about the effect of pH on binding oxygen. So, and this is called the Bohr effect. The Bohr effect is how pH changes the T to R transition in hemoglobin. So from this graph, we can see that the more acidic you are, the weaker you bind to oxygen. And we can do this by looking at P50. So here's 50% bound. At a pH of 7.6, that's roughly 22 torr. At a pH of 7.4, let's say that's roughly like 35 torr. At a pH of 7.2, I don't know, like 38 torr, rough numbers. But you can see as the pH becomes more acidic, your P50 increases. So under acidic conditions, hemoglobin does not want to bind oxygen. Now, let's understand the reason for that. Why is that a thing, just speaking about how the body works? So we're going to go down to our little image down here and talk about what's happening. So when your muscles are working, one of the byproducts of glycolysis and the citric acid cycle is carbon dioxide. And carbon dioxide is a waste product, so we need to get rid of it. So carbon dioxide will leave your muscle and go into the blood. But carbon dioxide is a gas and is not soluble. It cannot dissolve in your blood that well. So to fix that, we have an enzyme called carbonic anhydrase. And carbonic anhydrase will change CO2 to carbonic acid and make your blood more acidic. So when you're working out, when you're using your muscles, the blood around your muscles becomes slightly acidic. Now, based on what we just said, when hemoglobin goes into acidic pHs, the the T state is favored. Right? Because on this graph, we don't want to bind oxygen under pH, high or acidic pHs. So that means the T state's favored the more acidic you get. What that means is that for hemoglobin, which will go to your lungs and take oxygen, when it goes to the muscle and it goes to this acidic pH, it will want to go from the R to the T state. It will want to make that transition. When it's in the T state, it will release oxygen. That oxygen is taken up by myoglobin in the muscle which can then go and deliver it to the mitochondria so it can continue the citric acid cycle and oxidative phosphorylation. Now, you don't want your blood to be like really acidic. So what that hemoglobin will also do at the muscle is that it will pick up a proton from that carbonic anhydrase and take it to the lungs. Now, in the lungs, what happens is that we want to breathe out carbon dioxide. So we take this carbonic acid, and the same enzyme works that we originally used over here to go to CO2 to carbonic acid. We are now saying carbonic acid plus a proton release water, make CO2 again. We breathe that CO2 out. So that is how, in a nutshell, one, we can move CO2 from your muscles to your lungs to breathe out. We just change it into a different molecule, then change it back right at the lungs. That also shows you how we can take oxygen from the lungs and deliver it to the tissues that are actively working, because those tissues are releasing CO2, making your blood more acidic because of that change to carbonic acid. And that's kind of just summarized right here. So tissues release CO2 and release a hydrogen or a proton. The hemoglobins take up this proton. That will stabilize the T state, release oxygen. Then to the lungs, where you have very high PO2, like 100 torr. Inside the lungs, it's like 100 torr of oxygen. You release that hydrogen because you're going into the R state because you're binding oxygen. That proton is released to reform CO2, breathe out CO2, then the whole thing can start again. Pick up that oxygen, go to respiring tissues, drop off oxygen, so on and so forth. So that phenomenon of pH influencing hemoglobin is called the Bohr effect. And that diagram on the bottom there is the relevance of the Bohr effect inside your body. Right, so is there any questions about the information I went over there, or the Bohr effect, or anything at all? All right, if not, we can move on. Now, the next molecule we're going to look at that can affect the binding of oxygen to hemoglobin is called BPG. The proper name is 2,3-bisphosphoglycerate, so we're just going to call it BPG. Now, what BPG does, let's draw my little hemoglobin again. So my 4 subunits. BPG binds in the center of those 4 subunits. That's what's being shown right here. We have some N-termini going towards BPG. We also have some positive amino acids. So we have a lysine and some histidines, all interacting with BPG, which is really negative, has these phosphates, has this minus charge. So BPG loves being in the center of hemoglobin, which has a lot of positive charges. And when BPG will bind to deoxyhemoglobin, or the T state, much better than oxyhemoglobin. In fact, if you bind oxygen and switch to the R state, BPG can no longer really bind, and so it's expelled out of the central cavity. And this is how you can breathe effectively at higher altitudes. And this is why when you go to a higher altitude and within the first week or two, you are constantly short of breath. That's all due to BPG and hemoglobin, which we can see in this graph on the right. So these numbers here, 0, 20, 40, 60, 80, 100, that's just the pressure of O2. And remember, YO2 is fractional saturation, amount of hemoglobin bound to oxygen. So we're just going to look at the red line for the black line for right now. And inside your arteries at sea level, the pressure of oxygen is 100 torr. And so all your hemoglobin is bound to oxygen at sea level. Then in your veins, the pressure drops to roughly 30 torr. So when you do that, you have a change in fractional saturation of 0.38. What that means is that if you had 100 hemoglobins in your arteries that are bound to oxygen, 38 of them inside your veins lose oxygen, while the other 62 still have oxygen. That's what that drop in 0.38 means. So that's sea level conditions. All right. Now, let's say you went to Denver or you started climbing a mountain, all right? And the pressure at 4,500 meters up in the air is inside your arteries like 58 torr. And your veins are the same. So when you go up 4,500 meters, if you have 100 hemoglobins, 30 of them now lose oxygen, while the 70 still have oxygen bound. It's not quite, that math isn't quite being honest with you, because actually you shouldn't have 100 anymore. At 4,500 meters, it would be like you only have 90 hemoglobin to work with, but that's a very minor point. The point that's more important to get across is that at higher elevations, you are less effective at delivering oxygen. However, let's say you spend a couple of weeks at this higher elevation, which is common if you're climbing a mountain. If you're climbing like Mount Everest, what you do is that there's 4 stations along Mount Everest called like base camps. And you just practice climbing up to like base camp 1 to base camp 2. And we spend a couple of days at base camp 2, go back down to 1, 1 to 2, 1 to 2. Then once you're used to the elevation at base camp 2, you go 2 to 3, then back down to 2, 2 to 3, back down to 2, so on and so forth. And that actually has, we can actually explain what's happening on a biochemistry sense. So the longer you stay at higher elevation, your body will produce more BPG. And let's take a look at this. So the red line is higher levels of BPG after being at high elevations longer. So here, the amount of fractional saturation at a higher BPG is restored to sea level levels. Sea level levels, yeah. So here we have a 37% efficiency of delivering oxygen at higher BPGs, at this high level of BPG. So that's how your body will get used to being in a situation with less amount of oxygen, which happens at higher elevations. You produce more BPG, so you're better at delivering that oxygen rather than your sea level BPG amount. And why do we need BPG at all? If you didn't have BPG, you would never be able to deliver oxygen to your tissues. The hemoglobin would basically never let go. And so, and that's what's being shown in like stripped hemoglobin. If you look at the fractional saturation there, it really does not let go of the hemoglobin except at very, very low oxygen pressures, which we never see in the body. You never see like oxygen pressure being like 10 or 5 unless you're inside of a cell. So inside your veins, it doesn't get that low. So the hemoglobin would just stick on to that oxygen. However, BPG allows for that oxygen to be released by the hemoglobin because it wants to be in the T state. And just another use of BPG, right? So fetal hemoglobin, when compared to adult hemoglobin, fetal hemoglobin does not really want to bind BPG, right? So the hemoglobin of a fetus is different than an adult. What that means is that in adult hemoglobin, BPG will bind and release oxygen, which the fetus hemoglobin can take up more easily because it's not affected by the BPG as much. So there's another practical use of BPG when delivering oxygen to a fetus. Right, so with all of that information, is there any questions about BPG or what these graphs are saying or anything at all up to this point about hemoglobin? Right, if not, we can move on. So here is a way to test your understanding. So another mutation, Rainier, tyrosine 145 is substituted to a cis. When this happens, you form a new disulfide bond that isn't there normally. Here, with this new disulfide bond, you cannot stabilize the T state. Therefore, how does Rainier change with oxygen affinity, the Bohr effect, and the Hill constant? So I'll give you 2 minutes or so, 2 to 3 minutes to think about these concepts, what it means in the big picture, and I'll be back to explain it in just a few minutes. So take a stab at that, look back at your notes, try to remember what this stuff is, and I'll be back. All right, so hopefully you had some time to contemplate that. So if we cannot form the T state. We're always going to be in the R state. R state wants to bind oxygen, so oxygen affinity goes up. I want to bind oxygen more. The Bohr effect is when pH decreases, the T state is stabilized and you release oxygen. But if you can't go into the T state, the effect of the Bohr effect is decreased because you go down in pH, but the T state isn't stabilized, so the Bohr effect's not doing anything. And the Hill constant, so if you don't remember what that is, a Hill constant will tell you if you have cooperativity or not. So hemoglobin has a Hill constant that's roughly 3. And the way Hill constants work is 1 is no cooperativity. Less than 1 is negative cooperativity. When you bind something, you don't want to bind it again. Greater than 1, that's a pretty bad greater than sign, but greater than 1 is positive cooperativity. So if you only go to the R state, your Hill constant will decrease. It's going to go closer to 1 because remember at the very beginning of this class, we talked about that the cooperativity comes from that phase transition. It comes from that R to T switch that is felt from the other subunits. And if you never had that switch into T, the other subunits won't know if oxygen is there or not. So you have basically no cooperativity at that point. Right, so questions about that explanation? All right, so we can move on then. I believe, yeah, so this is my last question. So humans actually don't have a way to sense oxygen in their blood. When you are suffocating and that urge to breathe comes on, it's not because you're low in oxygen, it's because you're high in carbon dioxide. So that is what's really driving that. So you have no sensor of oxygen pressure. And keep that in mind. And so some free divers, and what a free diver is, it's those people who dive underwater without any gear. What they will do is before they dive, they will hyperventilate. So breathe in very fast like that. And they think that doing that will increase the oxygen content of their blood, which allows them to stay underwater longer. Does it do this? And use your knowledge of hemoglobin to think, is this a useful practice or not? Well, I guess I'll just go ahead and answer these. Short answer is no. Hyperventilating does not increase the oxygen in your blood. If anything, it decreases it. Because when you're hyperventilating, what actually happens is that you're making your blood more acidic. You're increasing the amount of carbon dioxide in your blood slowly because you're not fully expelling it with breaths, like normal breaths. You're not replacing it with oxygen like you would in a normal breath. Instead, you're hyperventilating, you're taking these very shallow breaths. And so your blood becomes more and more acidic as CO2 builds up. When the CO2 builds up in your blood and becomes more acidic, the Bohr effect is starting to kick in. And hemoglobin in your blood will give up that oxygen. instead of holding on to it. So what will happen is that you're actually releasing oxygen prematurely before you jump into the water. Rather, if you were in the water and you didn't hyperventilate, your CO2 would build up while you're underwater, and then the oxygen would be released. That's also like one of the reasons why you feel dizzy when you're hyperventilating, is that your blood becomes a little bit acidic and causes that dizziness. But that's what's going on a physiological level, and talking about hemoglobin and hyperventilating and levels of carbon dioxide and oxygen. It's really interesting once you get into the small details of how all these systems work, and you can start to just say on a chemical level, what is happening inside the body that we can see on the large, like macro level, like when a person passes out from hyperventilating. All right, so any questions about anything that we covered up to this point? All right, if not, that's all I have for us today. So I'll put a homework up like always. Just remember, test is on Friday. If you do have questions, feel free to email me. Otherwise, see you on Wednesday. Have a good rest of your day.